Good afternoon, everyone. And today, what we are going to learn is about dental pulp. Now, dental pulp, if you all know the structure of the tooth, uh, you very well know where it lies. It lies exactly in the center of the tooth, made up of living connective tissue cells, and they contain lots of cells. Um, their vitality of this cells is actually uh, very important for as a dentist point of view. That's what we are going to be learning today: the cellular structures, the histology, the development, to certain extent, not much, just where it starts, MCQ point of view. Then histology of pulp. Then um, you have structural features, functions, regressive changes, clinical considerations, and conclusions. I'll try to finish it within this lecture. Lot of things. So what is expected from your end is it will be a capsular, very fast learning about pulp today. So pulp have various signaling centers. Pulp has a different anatomy. Pulp is a very vital structure. It has a certain volume, and there are various canals. There is a pulp chamber, pulp horns, radicular pulp. So these are the various parts which will be discussed in a very fast forward manner, so that you are able to revise when you see the presentation again. So a rich, a richly vascularized, innervated connective tissue of mesodermal origin containing central cavity of a tooth, uh, delimited by dentine and having a formative, nutritive, protective function. Now, various functions, etc., we'll be learning later on, but this is some kind of a amalgamated definition of dental pulp. It begins at 8th week of intrauterine life. That is the key factor here you should keep in mind when you are learning about pulp. Now, pulp has a certain volume. I'll be telling you, in permanent teeth organ, it is 0.38 cc, and a mean volume of single adult is pulp is around 0.02 cc. So you have to keep in mind for viva purposes also. Now, this is the anatomy: cornal part, radicular part, apical part, uh, and accessory canals. Now, cornal part, as it says, it will be the crown part, the part which is within the crown. Uh, so that will be very important to understand that how do you divide the pulp. Now, keep that in mind. There is the cornal part is residing towards the crown part, and the radicular part towards the root part. This is exactly the cornal pulp development is near the crown of the tooth, and root is towards the apex of the tooth. Very simple uh, definition of this. You have to understand that there are various zones here. This is the anatomy of pulp, and then there are zones of pulp. Zones of pulp we'll discuss subsequently. We'll be discussing the cells of the pulp also. So general features being, it is located at the center of the tooth. 52 pulp organs are there. Volume, I told you what exactly is. The largest is the maxillary first molar, smallest mandibular central incisor. That means the volume asked in MCQs when it develops, what is a permanent size, what is a which is the largest one, which is the smallest one. Various layers of structural layers of pulp are there. The odontoblastic layer, the cell-free zone. Some people call it as an artifact, cell-rich zone, and pulp core, or the parietal layer of pulp. Now this, before going to the uh, discussing the cells of the pulp, you should know that there is the pulp core which is present, will have, is present at the pulp itself, is present at the center of the tooth, and within the center of the pulp is the pulp core. Now this has many cells, an extensive vascular supply. Uh, it is very similar to cell rich zone, but it is located in the center, I will say. And cell rich zones contain fibroblast, undifferentiated mesenchymal cells. This will be the cell rich zone. Then there is cell free zone, also called as zone of Wheel, WEIL. I'm talking extempo about all of them so that you understand how to go about reading about it. This is just a glimpse of, so that you are able to draw diagrams. If you see, 2 diagrams have been sent to you regarding the pulp stones as well as the zones of pulp. So these will be the 4 zones. One will be the pulp core. Then you have cell rich zone, cell free zone of Zeals, which is rich in both capillaries and nerve networks. So it is not actually cell free. That is, it is not a vacuum which is created. It is a misnomer, but just to tell a feature that it is, it is having capillaries and nerve networks. Odontoblastic layer is the outermost layer, which contains the outer odontoblast and lies next to the predentine and the mature dentine. So that's how is the setup is there. The cell found in the pulp include fibroblast, odontoblast, defense cells, etc., etc. Now you should know where is plexus of Rashkov present, which monitors the pain sensation. You should read about it. What kind of fibers are there? What are C fibers, etc., etc. When you read about it, I've just taken you to the plexus of Rashkov. You should know where it is present and how it is separated from the odontoblast by the cell-free zone of Zeal. So you have to keep in mind that this plexus lies between the cell-free and the cell-rich zone. Read about it. Talking about composition of pulp. Composition of pulp will be extracellular matrix, collagen matrix, crown matrix, and cellular components. Keeping in mind just this classification of composition of cells, we are going to discuss further. Extracellular matrix contains collagen and elastin. Then it was ground substance. Ground substance has glycose gags, that is glycosaminoglycans, proteoglycans, fibronectin, hyaluronic acid, chondroitin sulfate, water. Talking about cellular components of pulp, we had discussed that there are collagen and ground substance in the extracellular, and now in the cellular components, what you have is odontoblast, fibroblast, undifferentiated mesenchymal cells, and immunocompetent cells. Everybody's understanding? Guys, just type on the chat if you're understanding everything. If you want me to stop anywhere, just put your query so that I can go back. So talking about composition of the pulp, it was extracellular and cellular components. Extracellular components was collagen and ground substance. Extracellular matrix had collagen, elastin. Extracellular matrix had ground substance, which I have already enumerated to you. Then you have cellular component, odontoblast, fibroblast, undifferentiated mesenchymal cells, and immunocompetent cells. Talking about odontoblast, keep in mind you have to read about the structure and function, odontoblastic process, and odontogenic junctions. You have read about that in the dentine chapter, but just to tell you, you should know what it looks like, how the odontoblast look like, what are the cells present, how are they present, the structure and function you should know. Most distinctive types of cells are present. The cell rich supports the activity. These are, I have enumerated the in points. Go back, I've taken you to the book, read about it, how what exactly is the structure, etc. Odontoblastic process, length, how it looks like, what are microtubules, vesicles, how do they look like, what are the fluid present. Then are the junctions, gap junctions, tight junctions, or desmosomes. You have to understand this. So we have discussed odontoblast under three subheads: structure and function, odontoblastic process, and odontoblastic junctions. Read about them separately. Prepare a short note. Odontoblast is actually asked separately as a short note as well. So you have read about odontoblast. Now comes the fibroblast. Very important cells in this structure. It is committed to differentiation. It synthesizes the type 1, type 3 proteoglycans and uh, glycosaminoglycans. It is abundant in the cell-rich zone. It maintains the matrix, the influx of the cells, outflux. It is a thing which is maintaining the entire cellular setup, um, form and matrix of the pulp. So it has the ability to destroy even the phagocytose collagen. Also, it has capabilities. Then is the undifferentiated mesenchymal cells. These are the largely lightly stained abundant cytoplasmic cells. These are totipotent and found in the proximity of blood cells, cell rich, so in the cell rich zone and the core. So you have to keep in mind they are present at the blood vessels, cell rich zone, and the core. Read, keep it in your mind where exactly they are located. So this will be very handy. Then comes the immunocompetent cells. Each of them at least learn minimum basics about it because they are asked so very often about. So lymphocytes, macrophages, mast cells, dendritic cells. Lymphocytes present in the normal pulp and are also seen in the pulp of the impacted and the unerupted tooth. Macrophages, these are modified monocytes. They produce interleukin 1, tumor necrosis factors, then cytokines. These are very, very important cells, again present next to the blood vessels. They aggregate the vesicles. They are aggregates of vesicles or phagosomes. Now this is a little technical. What exactly happens here is that their location is for production of APCs. They are present as MHCs, class 2 antigen. They're active in endocytosis and phagocytosis. So they are present as aggregates of vesicles. They are present near, in short, irregularly shaped cells. They are present with short, blunt processes. So you need to really look for pictures available in plenty online. You will understand that these cells are very important part of pulp. So lymphocytes, macrophages, and then comes the mast cells. Mast cells rarely found in normal pulp, but found in chronically inflamed cells. So very important to understand that they are present in pathological cell rather than physiological features. So they contain inflammatory mediators like heparin, histamine, 1-HT serotonin. These all are inflammatory mediators. Then are the dendritic cells. They engulf the protein antigens. These are the cells which will engulf the protein antigen. Antigen presenting cells with the dendritic cytoplasmic cells are also present. The accessory cells of immune system. These are accessory cells of immune system. Again, these will be the least important after mast cells, but they are for a complete answer. You require all the 4 cells to be talked about, which are lymphocytes, macrophages, mast cells, and dendritic cells. So we have learned about various cellular components, that is odontoblast, fibroblast, undifferentiated mesenchymal cells, and immunocompetent cells. These are the cellular components of dental pulp. So we have read about most of the structures. Then is the pulpar vasculature. There are arterioles, venules, capillaries, lymphatic vessels. Need not learn much about them, how the fenestration happen, and what are the capillary basement membranes, what are the capillaries present, how are they present. Not much to, as a questions are asked, but then you have to keep in mind where they enter from, how they branch laterally, how they ramify into capillary plexus in the subodontoblastic region. These are to be learned about. Venules, lymphatic vessels, how the transfer of how the filtration and absorption takes place by diffusion, osmosis, and capillary filtration. So today we have learned much about the pulp. A lot of information was given today. What exactly goes on here we learned about. Before we go, I would like you to have a look at um, zones of pulp that has been taught today, because these diagrams have to be completed. So I will just discuss once again the zones of pulp. Everybody can see. Everybody can see.
Yes, sir.
Okay. So we have, if we turn the steering around, you will understand there is dentine on the top, which should be at the side actually. So dentine, then you have pre-dentine, odontoblastic layer, cell-free zone, cell-rich zone, and pulp proper. Any problem in this? Zones of the pulp, any problem in the diagram? None, right? Last year, you had read about pulp stones. So this will be the second diagram which has to be drawn. Everybody can see? Everybody can see?
No, sir.
Okay. Now everybody can see?
Yes, sir.
Okay, great. So we have the same diagram almost, but it is having different types of pulp stones. You had read about pulp stones ever? Read about them when you reach them. These are the concentric accumulation which are present in the pulp. If you want as a what exactly they are, what pulp stone means, and what where they are present. If you can see the picture, you see there is one embedded one, one is the free one, and one is the attached one. So very rightly, how is it is as per the location. These are also called as endolith. There are two types which are there, true and false ones. The true ones are formed by formed of dentine by the odontoblast. The false one are formed by the mineralization of the degenerating pulp. Often concentric patterns are seen in this. On the basis of location, this was on the basis of structure. The structure can be true pulp stones and false pulp stones. On the basis of location, it can be free, attached, or the adherent one, and the embedded one. So you can see, right? They can be present at the cornel or the root portion of the pulp in the teeth. They are not painful until unless there is a nerve impingement. So what you have is, on the basis of structure, it is true and false. On the basis of location, it is free, adherent, and embedded. Read about them, clinical implication, etc. Prepare a short note on this, and we are going to discuss next time what happens. So with this, we will proceed to part 2. If you have any doubt, you can post me. Thank you very much. That's